My name is Brian Dale. I'd like to talk to you today about the process of fertilization, which we're all involved with, a little about the theory, and then, of course, what we do in the laboratory. The process of fertilization is not well covered in textbooks. If you see most of the textbooks, they look at developmental biology, and perhaps fertilization covers a few pages. It's actually a very complex process that starts with the interaction of the sperm with the outer layers of the oocyte and technically ends up with the formation of the two pronuclei. On the slide that you can see on the top left, it's a scanning electron micrograph of the surface of a human oocyte, showing nicely the outer cumulus cells, the zona pellucida that's cut through in a wedge, and then underneath the plasma membrane. And of course, the sperm have to interact and go through all these layers before actually reaching the egg in a normal fertilization process. Before interacting with the accessory cells, the oocyte also has to be primed by the female reproductive tract, which changes a lot of physiology of the sperm as it goes through the reproductive tract. As it's going through the tract and also the outer layers of the oocyte, the sperm have to encounter signals that trigger changes in their physiology, which are prerequisites for subsequent steps. So in a stepwise process, it's going through the outer layers, and all the unsuccessful spermatozoa that do not encounter these signals will fall to the wayside. Fertilization is actually a physiological process of two cells already preformed, ready to interact with each other. The oocyte has to accumulate molecules of all sorts and structures that are laid down during oogenesis. And if oogenesis is suboptimal, we will not generate a viable embryo. So oocytes in most animals are metabolically repressed cells waiting for a signal from the spermatozoon to trigger it into metabolic activity and release it from the meiotic block. If the oocyte is not activated by the sperm, metabolic reactivation will always start in a slower fashion, and this is called oocyte aging. All oocytes from all animals are blocked in meiosis twice. First, in the ovaries, they're blocked in prophase of first meiotic division, and progression from this stage is triggered by hormones or ionic signals from the environment. Second block is actually in metaphase 1 or 2, depending on the animal species. The human, of course, is metaphase 2, where the sperm is responsible for triggering the resumption of meiosis. This slide shows the meiotic blocks in different animal species. As you see, they range from a block in prophase in the mollusks to a block at the end of meiosis in the sea urchin. Most animals actually fall in the middle, so we see the ascidians, they are blocked in metaphase 1 and the sperm releases this block. And the human and the frogs are blocked in metaphase 2. Again, the sperm is responsible for releasing this meiotic block. All oocytes are blocked previously in prophase anyway, as they are in the growing stage within the ovaries. So let's go back into what we do in the laboratory. So in the photograph, you can see a typical photograph of the human metaphase 2 lined up to be injected with a sperm. So intracytoplasmic sperm injection has really taken the majority of the techniques that we do in the laboratory at ICSI at the moment. So we can inject a sperm directly into the cytoplasm in the human. It's a lot easier in the human than other animals. In fact, in sea urchin, it does not work. In the mouse, it was very difficult to do. Actually, in the human, it's very easy to activate eggs by injecting the sperm directly into the cytoplasm. A technique fortuitously found by Palermo, I think it was in '92, about So we can actually bypass the sequential steps of cell-cell interaction, and in a way the selection or the morphological selection of the fertilizing sperm, and this does not impede activation, the formation of a viable embryo. This means that the molecules involved in sperm-oocyte interaction and their correct expression are not really necessary or correlated to the capacity of the sperm to activate the egg. Obviously, that doesn't mean they're not necessary. In natural fertilization, obviously the sperm has to deal with the outer membranes, the outer layers. So oocytes are often up to 1 million times the volume of a spermatozoa, and yet this tiny sperm can trigger the resumption of meiosis and all the metabolic changes that occur in the egg, the oocyte, and I'll explain those later. But generally, we need to understand that oocytes are programmed to interact with the sperm at a particular moment in time. And if they are interacting with the sperm too early or too late, it doesn't really work, and you get partial activation. So what is activation? The activation of the oocyte is a triggering from a quiescent cell to a metabolically dynamic zygote. It's quite a dramatic cascade of events that changes the whole physiology of the oocyte. The initial events are ionic, and these are translated into changes in the kinetics of cell cycle kinases, and this, of course, leads to the events of meiosis. Let's get on to the sperm. So spermatogenesis is composed of 3 phases: proliferation of cells by mitosis, reduction of the chromatin content by meiosis, and finally differentiation of the spermatozoa in a process called spermiogenesis. Basically, we end up with a cell that has 3 major roles to carry out, apart from the roles of penetrating the outer layers of the oocyte, which we talked about before, and that is down to the acrosome region of the sperm, which is either around the head, as in mammals, or right at the tip of the sperm, as in invertebrates. So basically, the sperm is what is delivering the chromosomes, obviously, but those are inert essentially at that point. The 2 other components are essential for the beginning of development of the centrioles, which the sperm carries, the division centers, and a sperm activating factor, which is a soluble chemical within the sperm that is necessary to start the whole process, the process of activation in the oocyte. This next slide, this cartoon, is really to show that not all areas on the oocyte surface are amenable for sperm entry, and not all sperm can actually enter and activate the oocyte. This is a cartoon from live videos from the sea urchin, and on the first figure on the left, you can see that after about 3 seconds, one of the sperm that hits the surface of the oocyte continues to gyrate around its point of attachment. About 12 seconds later, that sperm stiffens, the tail stiffens, and we know from other recordings, electrophysiological recordings, that at that moment there's a depolarization of the oocyte plasma membrane, and about 10 to the 10 cations enter the oocyte, and they're highly concentrated under the plasma membrane. A bit later, after 40 seconds, there's a repolarization. The successful sperm starts to be drawn into the oocyte, and then most of our information from fertilization is still from the sea urchin, and that's because we can get so many gametes from these animals. They're so easy to work with. and they can be experiments can be done at room temperature. The medium is of course seawater. It just makes it so much easier. So most of our information is actually still from this animal. So if you see on the micrograph, that's a transmission electron micrograph through the head of a sperm that's undergone the acrosome reaction, which is at the very tip. It's actually attached to the surface of the sea urchin oocyte. To get an idea of size, that's the contact point's about 1/10 of a micron square. The large granule on the left with the spiral structure on the inside is actually a cortical granule. So whatever happens, that the sperm factor must in some way pass through this acrosomal tubule into the oocyte and start the metabolic processes necessary for activation. The very first thing it does actually in this animal is activate a whole series of non-specific ion channels within the plasma membrane of the oocyte. And this is followed shortly by a release of intracellular calcium from intracellular stores that starts at the point of sperm attachment and spreads across the oocyte to the antipode. So mammalian sperm and the human sperm are actually designed quite differently from invertebrate sperm. They look very similar from the outside. It's just the construction of the membranes. So in the human sperm, as you can see in this micrograph on the right of the slide, the acrosome actually covers the whole of the head, basically. And there's a plasma membrane, an outer acrosomal membrane, and an inner acrosomal membrane. Now, as the sperm interacts with the cumulus cells and subsequently the zona pellucida, the outer and inner membranes, the outer acrosomal membrane with the plasma membrane fuses and releases the acrosomal contents, which are enzymes, a whole series of enzymes. And in the human and other mammals, this is a lateral interaction. It's not the tip of the head of the sperm that interacts with the oocyte. Presumably, after fusion of the inner acrosomal membrane with the plasma membrane of the human oocyte, there's the release of sperm factor into the cytoplasm of the oocyte. So these photographs show sea urchin oocytes that were activated with sperm factor, and these were the first experiments to directly show that a sperm factor existed, and we did this in 1983 in the States with Louis De Felice and Jerry Aronstein at the NIH in Washington, which was actually 10 years before the first baby was born with ICSI. So basically the top three figures, A, B, and C, show a sea urchin oocyte that's been microinjected with a sperm factor. So a soluble factor extracted from millions of sperm and then concentrated on a gradient, made isotonic with KCl and then injected into the oocyte. As you can see, it successfully causes the elevation of the fertilization membrane in the sea urchin oocyte, which is the result of the cortical reaction and demonstrates activation. Now, as a control in D, we just injected a sham isotonic KCl solution just to show that the injection itself did not cause activation. But as a double control in E, we then added sperm to that oocyte, and the sperm were able to activate the cortical reaction just to show that the cortical granules were still present. So in the 1980s, we tried to isolate the sperm factor, see what it was, and we slowly started to purify the factor, and we found out it had a molecular weight of about 10,000, as we thought at the time. Since then, the experiments have exploded in all animals, particularly in the mammals, And as you can see, there's many candidates for sperm factor. Now, a sperm factor is, as long as it's soluble and activates the oocyte, then it fills its role as the sperm factor. In the mammals, the quest has been to try and find a very specific molecule, and the most popular is phospholipase C zeta, which has been identified and also in the human. But there are also other factors in humans and mammals that have been identified, such as truncated tyrosine kinase and post-acrosomal sheath domain binding protein. And in the amphibians, there are certain sperm factors that have been isolated, such as citrate synthase, but even external ligand factors have been seen to be responsible for oocyte activation, which takes us backwards a little in the whole concept of how we activate oocytes. In the birds, again, citrate synthase was isolated, phospholipase zeta also. In the invertebrates, there's a whole range of potential activating factors, from calcium itself, which was suggested by Lionel Jaffe years ago, IP3 suggested by ourselves, cyclic GMP, ADPR ribose, and nitric oxide itself. So it's still very much open, and sperm factor could indeed be several of these molecules altogether. The first visible sign of activation in an oocyte is actually a small structure on the surface called the fertilization cone, which looks like a cytolytic zone of the oocyte where something cytolytic occurs, and that ties in with some very old observations of the turn of the century. In the photograph you can see these are fertilization cones of a frog called Discoglossus pictus, but it's very similar to ones you'll see in echinoderms in particular. So the first event in activation is actually a change in the membrane potential of the oocyte, which you can see in all animals in one form or another. In these graphs, we can see the fertilization potential, top left of a sea urchin, top right an ascidian, and the bottom graph is actually an amphibian, Discoglossus, I think it was. The depolarization can reach 100 millivolts, and we noticed in these recordings that each large depolarization was actually preceded by a smaller step-like depolarization. And this step-like depolarization was characterized by an increase in membrane noise, which indicates an increase in conductance across the plasma membrane. In the sea urchin and the ascidians, we estimated that the ion channels that were opened by the sperm, we call them fertilization channels, had a conductance ranging from 30 to 400 picosiemens. In the mammals and in the human also, the first event is still electrical, but it's actually an event that causes a hyperpolarization of the membrane rather than a depolarization. And the first event in the human is actually due to the activation of calcium-activated potassium channels, which are very common in the human body in all cells. And we discovered this in '95 with working with Gianaroli in Bologna. And the top frame on this particular graph shows what happens at activation in the ascidian. So the very top series of photographs shows the contraction of the oocyte. The middle graph shows the inward current that's generated at fertilization, which reaches about 1 nanoamp, which is quite large. And the bottom graphs show the calcium wave in a fluorescent microscope traversing the oocyte. In the bottom frame, we can see human M2, perfect M2, at a controlled temperature with controlled pH, and it was fertilized with a microelectrode in it. And you can see that the depolarization is, the current is outward, and that's due to the activation of calcium activated potassium channels. The ubiquitous activation signal is actually the release of intracellular calcium within the oocyte. The calcium is stored in intracellular organelles such as the endoplasmic reticulum, and more recently we think the actin cytoskeleton stores calcium. There's a very large gradient from the organelle into the cytoplasm, and the sperm, you can see on the graphs on the left, hits the oocyte and starts this calcium wave, which you can see in red, which passes slowly across the egg until it reaches the antipode. Now, this release of calcium needs a large energy, a large amount of energy to be able to do that, and probably oocytes differing quality owing to the size of this calcium wave or the energy necessary to release the calcium wave. The calcium wave is actually triggered by the release of intracellular second messengers such as inositol triphosphate, and that is released by the sperm. And as I say, it starts at the site of sperm entry and traverses the oocyte to the antipode. These two video clips show a fertilization in the starfish oocyte. The left is the right image is a higher magnification of the left. You can see the sperm coming to the top left corner. The oocyte is surrounded by a thin fuzzy layer, which is the vitelline membrane and the jelly coat. And the sperm hits basically the jelly coat, pushes out a long tubule, very long tubule in this animal, the acrosomal tubule that goes right through the surface of the jelly and the vitelline coat and hits the membrane and activates the egg. As you can see, there's a huge calcium wave shown in yellow that traverses the egg from that point to the antipode. The details you can see better on the clip on the right that show the blue, the sperm color blue, because it's fluorescently labeled with Hirsch. You can very clearly see this tubule pushing through the outer layers, and so activation actually occurs before the sperm head actually gets close to the plasma membrane of the oocyte. And as a result of the activation wave, the calcium wave, there's a contraction that you can see around the oocyte, and also the elevation of the fertilization membrane at the very end of the clip. Calcium waves are found in all animal oocytes at fertilization. They differ a little in their speed and the type of calcium waves. In some animals, it's one large calcium wave. In others, such as the ascidians and the humans, there's a series of calcium waves that traverse the oocyte. Now, in the ascidian, in the figure on the top, top panels on the figure, you can see the contraction, photographs of the contraction of the surface. The middle graph shows the calcium waves that come in one very large calcium wave followed by a series of oscillations. Then it stops between the two meiotic divisions and starts off again towards the end of the second meiotic division. And this is correlated with the inactivation MPF, which are the cell cycle kinases, which you can see on the bottom graph in yellow. second structural event of activation that you see in most animals, or if you don't see it, it actually occurs, is the cortical reaction. These four frames are taken from the sea urchin, uh, and it shows the elevation of the fertilization membrane, which is actually the expression of the cortical reaction. So the cortical granules are lots of little granules of about 1 micron at the surface of the oocyte. They're also found in the human. The sea urchin is about 20,000. They need to be subcortical, otherwise the oocyte is not cytoplasmically mature and will not activate correctly. So the top left shows the moment of fertilization in the sea urchin. The sperm is actually hitting the egg. The successful sperm is on the lower left. Now, if you go to the frame top right, you can actually see the sperm. The surface of the oocyte is a little flattened, then you can see the little blue sperm, again colored with the fluorescent color Hirsch, halfway going into the oocyte, and the membrane is elevated around the sperm. Bottom left, you can see the membrane elevated even further as the reaction has gone right round to the other side of the egg. And the last frame, bottom right, shows the fully elevated fertilization membrane in the sea urchin. This is a schematic representation of the cortical reaction and exactly what happens in the sea urchin. This is taken from my 1973 book, Fertilization Animal. So you see the granules are right up against the plasma membrane. And what happens is they are drawn closer to the membrane. They actually expand. There's a fusion between the cortical granule membrane and the plasma membrane, and the contents are released. Now, these contents then attract water within the perivitellin space, which causes the membrane to elevate, and you're left with an elevated fertilization membrane. And another component called the hyaline layer actually sits onto the plasma membrane. So let's get back to the human, or at least the mammal. So the most information or identification of a sperm factor is actually in the mammals. It's been studied quite a lot in the last 20 years. It seems to be phospholipase C zeta type of phospholipase. It certainly causes calcium oscillations and the release of inositol triphosphate if we inject this substance into eggs. It's found in mammals, birds, and fish. And if the sperm in a mammal model are negative for PLC zeta, they fail actually to trigger the calcium oscillations, and you get polyspermy. The negative thing is that we found out in IVF, human IVF, that if there are PLC zeta deficient sperm, they can still activate oocytes, and the males are subfertile. This was found out in mouse IVF, suggesting that anyway, if the sperm do not have PLC zeta, there are alternative pathways for activation, such as inositol triphosphate or calcium itself. And it's good to remember that sperm contain enormous amounts of calcium and inositol triphosphate anyway, so they would have enough of these chemicals upon fusion to activate the oocyte anyway. So the most complete story of oocyte activation to date is the work we did in the Ascidian in the 80s, 90s, and 2000s. And this is graphically shown in this graph here. So the sperm hits the plasma membrane, fuses, sperm factor goes into the cytoplasm of the oocyte, and has actually a dual function in the Ascidian. Through the production of nitric oxide and ADPR ribose, the fertilization channels are opened, and there's an enormous influx of calcium from the external medium, which is seawater. The sperm factor has another effect. It breaks down PIP2 in the membrane, producing inositol triphosphate, and this inositol triphosphate releases calcium from calcium stores, and this is the signal that eventually gets to cell cycle kinases and starts the cell cycle off. When we talk about meiotic resumption, that's the whole point of fertilization. We are talking, of course, about the cell cycle, which is very similar in meiosis and mitosis. Meiosis is a little different. For example, there's no checkpoint in certain areas because of the double duplication of the genome. But anyway, checkpoints ensure the correct control of events before progression. So we can imagine the amount of important pathways in this process that can go wrong and lead to an abortive meiotic resumption. So when we talk of cell cycle kinases, basically we're thinking of two factors: maturation promoting factor and CSF. Now, maturation promoting factor, these were both, by the way, discovered by Masui in the 70s, working on frog oocytes. And then since then, everybody's realized that they are the same factors controlling the cell cycle in mitosis in old cells. So the MPF is the activating factor, which maintains meiotic arrest because it's present in a high state. And this high state is maintained high by the presence of cytostatic factor CSF. Now, cytostatic factor is made up itself of MAP kinase and the product of the CMOS proto-oncogene. So again, when we think of meiotic resumption, we should really imagine in our heads the complexity of this process, the topographical and temporal complexity, and how easily things can go wrong and lead to a bad meiotic resumption. As I said before, CFS is mainly composed of CMOS and MAP kinase, possibly other kinases such as CDK2. MPF, maturation promoting factor, is composed of P34CBC2 and a regulatory subunit family of proteins called the cyclins. So again, let's look again at what we're thinking about in the laboratory. So we all know that human oocytes decrease in developmental capacity with age. But even in a young lady, there's a large difference within a cohort. This is presumably due to cytoplasmic maturity. So we see all the oocytes are in M2, but that really doesn't mean anything. They are very different in their capacity to go forwards and in their content, the quantity of various products within the oocyte. Maternal age is really similar to the free radical theory of aging. And it's probably an energetic problem, mitochondrial problem. In cells anyway, there are two respiratory pathways, cytoplasmic and mitochondrial. And it's been estimated that 50% of respiration in the human oocyte is in fact aerobic. And then of course Van Blerchem in the 90s showed that only human oocytes with a very high ATP content of more than 2 picomolar had implantation potential. In the 1990s, we did a lot of work with Martin Wilding from our laboratory in Naples on mitochondria, staining them with fluorescent stains to show the distribution of mitochondria and also the activity. And this is an example of differences we would find in human oocytes that we would not notice just from a light microscope. So the 4 frames on the left show 2 M2 oocytes, which would look completely similar. If you look at them in fluorescence to look at the distribution of mitochondria in particular, you can see that the top oocyte has a topographical distribution. So there's these patches of mitochondria, and these are actually these asymmetrical oocytes are the best and lead to and have a high implantation potential. Now the graph on the frames on the right show another 2 M2 human oocytes, now stained with a dye, looking at mitochondrial activity. And you can see at the bottom oocyte the activity, which becomes, when it's very high, is red. The nucleus of the M2 is actually a bit messed up, and the chromosomes in red. The very right bottom hand frame are disorganized compared to the top right hand frame, where you can see the human chromosomes in red on the spindle in green, and they're lined up on the meiotic spindle. So there are many chemicals in our world today. Many thousands have been produced since the Second World War, hundreds of thousands, in fact. Many of them are detrimental to us and to the quality of oocytes. A1254 is a polychlorinated biphenyl, which is very common in industry, and we get exposed to it as we get exposed to dioxin or bisphenol A. And all these chemicals act as endocrine disruptors, and they can change the distribution of organelles in human oocytes, in all human oocytes. And essentially, particularly the relocation of mitochondria during maturation, which is due to the cytoplasmic cytoskeleton. Obviously, if we have these mitochondria distributed in the wrong way, we're going to have incompetent oocytes and not get embryos. So this last graph is actually just showing us a cartoon of a human oocyte and the complexity of the what's inside it, the Golgi, the mitochondria, the cortical granules, the spindle. They're all attached and connected to the cumulus cells through gap junctions. But anyway, just to remind us that yes, we can have nuclear maturity, the chromosomes are aligned, it really doesn't mean anything. This is a particularly good signal to many medics who think doing a pickup and getting hold of an M2 is the end of the world. It's not. Cytoplasmic maturity is what we're interested in, and it's not always synchronous. On the left, there's a whole range of components of the cell, and then towards the bottom, when we think of the complexities of the cell cycle kinases, all phosphorylation processes, what glutathione's doing, and the ion fluxes and gradients, and even water, we realize how little we know about oocytes. So to conclude again, so there we are sitting in the laboratory with 10 M2 human oocytes in front of us. They all look very nice, but how do we get a viable blastocyst? Which one's going to give us a viable blastocyst? So in the oocyte, complex cell, very important, the topographical and quantitative information in them, and certainly not all the oocytes in a cohort are similar. They look similar, they are not similar. The sperm must have a good quantity of sperm factor to kickstart the oocyte into meiotic resumption and metabolic activity. Not all sperm have the same quantity of sperm factor. Oocyte activation involves ionic structure and biochemical changes that lead to the resumption of meiosis and the formation of the prenuclei. Extremely complicated. Again, it's a geographical problem. If it doesn't occur correctly, you're not going to get a blastocyst. So all of these prerequisites and events may be suboptimal, leading to abortive development. And in all cases, we require an adequate energy source in the oocyte. Thank you.